Okay, so here we have the Calvin cycle, and the whole point of the Calvin cycle is to produce one molecule of glyceraldehyde 3-phosphate. Where this process starts is at the 3 molecules of ribulose 1,5-bisphosphate, and this comes over here and it joins up with 3 molecules of CO2, and from there we get 6 molecules of 3-phosphoglycerate, and then it is required for 6 ATP to come in, and they end up becoming a 6 ADP, but that converts this to 6 molecules of 1,3-bisphosglycerate, and from there we have 6 NADPH that come into the reaction, and it turns out to be a 6 NADP plus an ion molecule, and then from there it goes down to 6 molecules of glyceraldehyde 3-phosphate. 6 molecules of here, 5 of these move on to this step, 1 of these branches off to this 1 molecule of this thing. This is the whole point of this, is to produce this molecule right here, and then the rest of these 5 keep going up, and it becomes ribulose 5-phosphate with 3 molecules, and then there's 3 ATP that come into this, and that turn into 3 ADP, which converts this back to where we began. In this overall process, it requires 6 ATP here, 3 ATP here, so 9 ATP in total to actually get from 3 molecules of CO2 to 1 molecule of glyceraldehyde 3-phosphate. On the other side, we have the light reactions. In the light reactions, we have a photon of light right here that enters the photosystem 2, and from here it enters, and then there's an electron. The electron is taken down over here, or it's converted to a negative charge on this side through the photosystem 2, and then we also have an enzyme right here that takes two H2O and breaks it into oxygen and 4 hydrogen molecules that are ionated. From there we go to the plastoquinone. From there we also go to the cytochrome, and then it goes to the plastocyanine, and then it enters to photosystem 1. And over here there is also a photon of light that is absorbed into it. The same exact thing that happens here with photosystem 2. It sends it to the negative charge over here, and then from there it goes to ferredoxin, and then it becomes ferredoxin NADP reductase, and then from here we have NAD plus ion plus a hydrogen ion, and it forms NADPH, which this is the whole point of this process right here, is to form this molecule right here. And that is all.